Into my life. Now I'm gonna do crack. It's fine. It's fine. And I have the tea kettle at home because I brought it home. Yeah. And then everybody full the sign sheet.
That's a new one.
For 86. Right, because I had like in the words.
Sometimes you just need somebody in the same room to keep you honest, you know.
If you don't mind starting the recording, that'd be swell. Hello, everybody, and thanks for coming in today. Dr. Sam Shepard is Dr. Sam Shepard serves as a health scientist in the informatics group within the CDC Influenza Division's Office of the Director. He has master's degrees in applied math and computer science from Bowling Green State University in Ohio. And before he wrapped up a PhD in biomedical sciences with a certificate in bioinformatics at the University of Toledo in Ohio. After graduation, Dr. Shepard went to the Fachhochschule in Salzburg, Austria, as part of the Marshall Plan, as a Marshall Plan scholar, and he joined the CDC in 2011 as an ORISE fellow. He became a full-time employee at the CDC Influenza Division in 2013, where he contributes to global influenza surveillance, supports vital program functions, aids the CDC's COVID-19 response, and provides mentorship to graduate fellows. He was my mentor at the CDC while I was an ORISE fellow, and helped provide exceptional guidance as I transitioned to from financial to biology. Dr. Shepard was one of 2 associate directors for informatics from 2019 until recently, and is a 2017 Charles C. Shepard Award winner for his work on the Iterative Refinement Meta Assembler, IRMA, which is a tool for next-gen sequence assembly of viruses. Thank you, Sam, for taking the trip down to visit and teach us a little bit about your work at the CDC. Please join me in welcoming Dr. Sam Shepard.
Well, it's great to be here, and yeah, I'm Sam Shepard. It should be said that any opinions you may hear today are my own and do not represent the policy of the United States government. So just want to say that to cover myself a little bit. Yes, I've been working in flu for pretty much my whole career at CDC, but lately I've had the pleasure of also simultaneously working on COVID. So say we all. So I want to talk a little bit about virus surveillance today, which is a topic near and dear to my heart. And the first word in virus surveillance is a virus. As you probably know, viruses enter our cells, replicate efficiently and quickly until they are finally, hopefully, stopped. And for RNA viruses, which is flu is, you can have mutations every replication cycle. So instead of a monolithic virus, you actually have a virus swarm swimming around there. You get sick, you go to the doctor, you get swabbed, cases reported, and maybe a specimen gets shipped somewhere. In the US, we have influenza reference centers that share sequencing load and send data or specimens to CDC. You can see one in California, New York, and Wisconsin. They can act as catchment areas for other state public health labs, which there's one in every state. Around the world, this model is kind of replicated. This is the global influenza surveillance response system, and all these little triangles are so-called national influenza centers that are responsible for local surveillance. So they may submit on behalf of other labs around the area. For this talk, we'll share information from more of a lab data perspective. The case reporting from hospital networks and all the stuff you probably saw happening on the news about COVID, the epidemiology is very important for that sort of work, but it's not my specialty, so I'm not going to talk as much about that. So on to sequence surveillance. Consider flu. I think probably one of the most deadly viruses in 1918, influenza kills 50 million. There's been multiple pandemics since then. There's always the threat of a bird flu, which is extremely lethal, is threatening all the time. We want to track the virus's evolution, mutations of concern, and that starts with sequence assembly. And for the genome, for flu, we are dealing with the segmented viruses, so each gene can fit on a different gene segment. Each specimen could be a lot of different types, subtypes, lineages. This amounts to a bit of reference juggling if you go with the traditional sequencing and assembly approach. And flu is always evolving, always. It's very diverse. We're doing hundreds of isolates with 8 segments every week, and each could be a variety of different types, subtypes, and lineages, and so that's a lot to juggle. Or maybe perhaps you get a virus that somebody had in a freezer that actually happened, and you've not seen anything quite like it. So that's the kind of the context for the work that I did on sequence assembly. So as mentioned earlier, this was the IRMA project, which basically we needed to deal with influenza's diversity in a high throughput way without involving humans as much. Because obviously that's the point of slowdown. Solution is iterative refinement, which is something you've probably seen in your classes and probably use every day. The sequence surveillance starts before assembly with lots of important work done by the lab for RNA extraction and cDNA and library construction and actually running the machines that do the assembly, the sequencing. The portion where I sort of fit in is this portion on the bottom. It covers a whole bunch of different steps that are necessary as well. To assemble flu, we assess quality. I won't go into that. And then we have 2 iterative refinement phases for dealing with flu's diversity. There's an iterative read gathering step, and there's an iterative final assembly step. You can think of read gathering as a 2-bucket system where you have reads in an unmatched bucket being moved to a matched bucket for particular genomes or gene segments. Reads are matched and moved to what we are specifically targeting. But as mentioned, flu is segmented and has multiple types and subtypes, and so we have to classify or sort into a variety of buckets actually. And so some buckets have lots of data and some have a little, and if they're 2 of the same gene segment, which could be contamination, you have to pick one, so we typically pick the stuff that gets the most signal. Now in the real world, we don't know the consensus ahead of time. And instead of selecting from a panel of references, we pick a reference and hope that we can move it towards our reads. I call this a reference seed or initial reference, which is in this case a very consensus from a very large multiple sequence alignment. We could have picked other things like a clade representative, but it's worked well in practice. The first step is to match our data to the references, sort them into our buckets, and generate a rough alignment for each of those. This is called reference editing. Although generally we try not to throw away reference as we go along. We look at our unmatched read pool by matching against the now edited reference, find more reads to match, and this increases the size of our read pool. We do this again and again until we either have no more reads to match against or until we have some sort of cutoff. And now we have gathered our reads and we have a candidate reference that matches them. And so if the first step is to sort of maximize the number of reads that match our different subtypes and gene segments and lineages, the next one is that we want to maximize the actual alignment score using something like Smith-Waterman, which I'm sure you could all write out. Nice recurrence relation for me. We try a series of changes. If it helps, we keep editing. If not, we stop, to put it simply. These are governed by various rules and heuristics. And so you can do things like mutate, insert, delete reference at this point, because you don't, sometimes that can happen. And as before, we move the reference towards the reads, maximize the score, but different from before, the read pool here you can see is it's constant. Now we're just maximizing the score. And then when we're done, we're ready for things like consensus generation, calling SNPs, or really we call them single nucleotide variants, look for variant phasing, generate tables and figures, and all sorts of other intermediate data. There's a lot more to say about this topic, but you can probably read that online if you're really interested and find the paper. So now I want to move on to the next topic. We have a bunch of sequences. What do we do with them? So it makes sense that we want to group them genetically for downstream analysis, for comparison, to compare contrast. Creating genetic groups is usually done with a phylogenetic tree. There's as much art here as science, how you lump or split different groups. The most important thing actually is that everybody agrees on what those groups are, which may seem counterintuitive to you, but it's actually the truth. I worked on a program called Label that does plate annotation using hierarchical classifiers. And in my opinion, as you get more and more detail and requirements for a particular process, your solution begins to mimic the data itself. That's just sort of my own anecdote. To make a long story short, Label takes queries, scores them against profile hidden Markov models, and then classifies them recursively using support vector machines. There are lots of other classifiers and techniques like NextPlay that we also use at CDC, and you can lump them all together and chop things different ways. I mentioned Label and earlier Irma because they are my work, and we do use them at CDC, but also because I got help from a jolly man with a beard that you might know. Not that one. Oh yeah, that's the guy. Justin Ball, he's a great collaborator. So until now, I've talked mostly about things related to genotype, but we have another area that's very important. Virus antigenicity. In other words, how the virus proteins, their molecular shape, affects their detection by antibodies. The virus contains the antigen, while the antibodies can be isolated from blood serum. If you get exposed to some virus, your body generates antibodies to it. Simple enough. Now you have antibodies that can bind to the antigen. But actually, you probably have multiple different kinds of antibodies that you generated. And some of those will be weaker or stronger in their affinity. Now, for routine analysis, it makes sense that you use a naive animal to try and get blood that has just been exposed to the virus of interest. And now you're ready for an antigenic assay. You have your viruses, some kind of cell for the virus to act on, and panels of sera containing different antibodies. I don't know if any of you have seen something like this before, but this is a classic antigenic assay called hemagglutinin inhibition. Hemagglutinin is the surface protein for flu. The goal is to measure when antibodies go from stopping the virus from sticking the cells together to letting them clump. And so there's a serial dilution involved. The other types of tests to measure antigenicity, but they're all about trying to stop the virus's activity on some sort of usually cell via dilution of antibody. So from the assay, you get a table of titers with columns of sera raised to different reference viruses, say a vaccine or different clade representatives. And for the rows, you have viruses that are circulating that you want to test against. And for the columns, you have that panel. And you may realize this already, But broadly speaking, if you have features of for each test virus, you can manipulate these until you have a distance matrix. And if you have a distance matrix, why, depending on the analysis you want to do, you can look at it a lot of different ways, whether from scaling the data, looking at a heat map, doing nearest neighbor, or even creating a tree with distances. But consider this distance data just as an example. For the same set of viruses, you can build heat maps from both pairwise genetic distances, so maybe p distance, maybe RNA or some other evolutionary distance, maybe from amino acid difference, physiochemical distance. And then, of course, you can take those titers and build antigenic distances. And you may notice that relating the antigenic and the genetic information for the same set of data is not one-to-one. It's not always clear, cut, and dry, even for very high quality data. It's more nuanced. But we do try. Here's another case where we're looking at distance data, and this is for antigenic data for both sides and the same set of viruses. But in this case, we have an avian host that we're raising Sp23 chickens, and on the left you're having actually ferrets, mammals. So even using the same set of viruses to inoculate, we can get clade-specific variations depending upon how we're raising those antibodies. And of course, we have to continuously surveil virus antigenicity, and so for flu, that usually means ferrets for our animal model. And if you're wondering, a ferret is not a human. We're not the same. Have you ever seen them? They have lots of sharp, pointy teeth. So when it comes time to pick a new vaccine, it's not like we can find a bunch of immunogenically naive humans, inoculate each of them with different viruses and hope that will work. Instead, we have to rely upon human serum pools from different populations around the world. And generally, we're trying to ask the question, when we test various circulating strains, is the antigenicity as good as our vaccine virus? So this assumes that we have persons that can give us blood before and after vaccination to the current vaccine. So we set a goodness cutoff and compare the vaccine antigen and our representative strains. And if the reactivity is good, great. If not, we have a bad vaccine match and probably have to change it. And so we can do this across different populations and age cohorts to get a better picture of what's happening around the world, at least antigenically. Vaccine selection is actually a very holistic process. You can almost think of it as like a big workshop involving WHO collaborating centers. And it combines the very best epidemiology, genetic, antigenic, and even structural data that we have from a whole bunch of different places, very smart people. You want to pick a vaccine strain that can be used to offer good protection to whatever will be circulating widely in the coming year. And as you know, there are multiple viruses to consider, not just flu A, but also flu B, and not just flu B, but also Yam and Vic, not just flu A, but also H1 and H3. And so there's a lot of clades then within each of those. And so it can be complex. But we don't have a crystal ball, unfortunately. It broke. Someone kicked it. So we must recommend a vaccine early to be ready for the actual flu season. And I think that's something that people don't realize is that there's a whole bunch of months in between making a recommendation from WHO and actually having vaccines available for you to get your shot, at the start of the flu season. There has to be ramp up time. And between those times, things can change in terms of what's circulating. And it should be also mentioned, because I think this is a little quirk that I think interesting at least. There are actually two vaccine selections that go on. There's a northern and a southern hemisphere. As it turns out, next Monday, they're starting the northern hemisphere vaccine selection. So that's happening overseas. And at WHO, CDC will participate. And weather, as you know, differs between northern and southern hemisphere, and so does the flu season. So that's why we have these two different vaccine selections. Okay, so we've talked about sequence surveillance, we've talked about plate annotation, we've talked about antigenicity. This topic, I think, is probably understated in grad school a little bit. Maybe not in some of your labs, but just when I was going to grad school. Undergirding a lot of the decision-making and things that happen in public health and virology are analytics capability. So this is making decisions powered by data. And to be able to make good decisions, you have to integrate data, query it, explore the patterns that exist. I'm mainly going to focus on lab data, but this is equally relevant for the epidemiology. So the role of traditional relational databases is important. We have them usage for transactional systems, for laboratory information management, other buckets of different information. These systems start to kind of get weak in the knees when your data get large, which is true for genomic data. You can ask Garrick about his experience later. We can overcome this using distributed storage, distributed compute capacity. We can ingest data from different sources, flatten it, analyze it at scale. And there are a lot of data domains to consider when integrating data. So flexibility and robustness are extremely important. I'm just going to pause and let you look at those words. It may not be exhaustive. If you're doing your job right, this is an unfortunate fact, others may not notice what turns out to be essential to the whole process, which is clean, standardized data. It's kind of like the pipetting of bioinformatics cleaning data. And a data model that makes querying things both natural and coherent. So, pop quiz. If you're at CDC, starting out, do you need to know R or Python for data engineering? No one's gonna venture a guess. Well, the correct answer is yes. And also SQL. So learn everything you can. If you're like me, I don't know if you guys are big Linux fans. You might have worked a little bit like this, where you're dicing data up and sorting it and running things through multiple different scripts and trying to pick out patterns and predicates and all that good stuff. I still do this. However, when you get a well-modeled database, you can find that you can reduce your work substantially using a query, which does all of those types of transformations, projections, aggregations for you. And then once you get hooked on doing that, you start working in your little query editor and answering questions that way. And then, oh, I'm going to connect my BI tool and I'm going to create visualizations from that. And then if you're like one of my colleagues, you're going to integrate everything and display it at the weekly meeting, and a lot of information there. I can go into it. So the point here is that once you take the trouble to really manage your data well and host it in distributed query engine, you can use it however you want. Query engine lets you prepare your data for downstream analysis use cases, perform more advanced analysis. I kind of mentioned this before, but there's just a lot of different kinds of I would say even mathematical operations, but also computational layers that you can add to your data. So it's whether it's a script or visualizing or making even more data sets, doesn't matter as long as you have a strong central data store. We like using query engine so much, and Garrett can attest to this, that we write custom user-defined functions and aggregates and libraries to avoid repetitive tasks that would otherwise exist in our scripts. That can be populated, that would otherwise have to be pushed downstream. And this is actually useful because a lot of different people can connect to your data. So let me try to put this concretely. You can put a lot of application logic on top of a a query engine. So say you have antigenic and genetic sources, you can apply your statistics, create business rules, develop from exploratory analysis, and then make decisions about where to put your precious lab time based upon some analysis all in your database system. And I tried to allude to this earlier, but there's a lot of different roles that will access that data in different ways. And so if you have a good, strong analytics practice in your team, you can democratize that data, democratize those analyses. So now you may ask yourself, that's great. I like research. Why public health? I also love research, and the questions I now ask are probably focused by and refined by the mission that I have been given at CDC. Public health is impacted not just by advances in science and technology, but season by season, outbreak by outbreak, need by need, emerging infectious disease by emerging infectious disease. Our mission and our scope can change just like that. This is off my notes, but I went from working on flu to working on flu and COVID, and taking the things I had learned in flu and applying them to COVID, and then taking things I did in COVID and applying it back to flu. And sometimes there's nothing like it. And I don't know, I suppose it's sentimental as well, but having a good vaccine, being able to understand whether or not strains are becoming drug resistant, developing cross-cutting approaches that can help with multiple diseases. I think these all can make a real impact and a real difference. So it's something to consider as you think about your future prospects. Which leads me probably to a question that you already have that I'll answer now. Maybe you want to get involved in government work. And so there are a lot of different paths that you could take. There are contracts that do business with government contract companies that you can find, and they'll tell you sometimes they're doing business with CDC. But their work can be more seasonal or ephemeral depending on the contract. There are fellowships which are more academic. They're more about training and research, like a postdoc. And Garrett can tell you about his personal experience with that. It was a very special time at CDC with the pandemic. They're a really great way to get your foot in the door if you're interested in kind of wading into it. I was an ORISE fellow, for example, and I decided to stay somehow. And then if you're really, I'm ready, you're rolling up your sleeves. You could try USA Jobs, which is where you can find a permanent position. And those can be quite competitive, but they're open to anyone who wants to apply and has the background. All right. I think I covered a lot of different topics, kind of broadly and high level, but I'm happy to answer your questions about things in particular. Yes.
So besides flu and COVID, are there any specific divisions at CDC that look at traditionally neglected viruses?
So you're asking, are there other divisions that look at other viruses?
Yeah.
All kinds, yes. So things get kind of divided up, I won't say arbitrarily, but along different lines. So there's an entire center devoted to immunization and respiratory diseases. There's another entire center devoted to emerging infectious diseases. And then there are other things that are non-infectious disease entirely that are also looked at by CDC. And so just within the respiratory immunization center, we have a division of viral diseases. We have an influenza division. We have a division of bacterial diseases, which are respiratory. And then very recently, I don't know if I'll say this on the DL, we just got approved for coronavirus and other respiratory virus diseases, which means that there's still more viruses being focused on. So depending upon the outbreak or the need, CDC will reorganize itself to try and look at different types of pathogens. And sometimes they're at the branch level or the team level. Sometimes a division might handle multiple different viruses, or maybe there's an entire polio group or entire group related to foodborne. So it just depends on the particular one. But yeah, it's we probably have a group for everything. So hope that answers your question. Yeah.
So I know it changes by season, but what percentage of your week is communicating with other teams and just doing science communication in general?
So with other teams within my division,
Or just in general, how much of your week is spent? Yeah.
Since the pandemic, I do it almost every day because I collaborate. I do two jobs essentially. I work on COVID and flu, and so the people who are moving into that new COVID division, I work with them, and I work with the different groups on flu. I sit in the office of director for flu, which means I work with our different branches. So we have an immunology branch, we have a lab branch, we have an epi branch, and now I think we have a new global branch. And so we can work with any of those branches where we sit as informaticians. But then within each of those, there could be data analysts, data scientists, other bioinformaticians. So, informaticians can be embedded at different levels of the hierarchy, and you communicate with all of them at different points. But yeah, I would say at least 33% of my time is spent communicating and going back and forth and reviewing other people's work. Yes.
I have a really dumb philosophical question. Would you be okay if I ask?
Yes, please.
So biology 101, my first biology class, they said that viruses technically aren't life. With someone like you who works with viruses all day, would you agree with that classification?
It depends on what you consider to be alive.
Yeah.
Do they replicate? Yes. Do they evolve? Yes. Can they do it on their own? No. So do they have encapsulation? Yes. Do they rely upon someone else's metabolism? Yeah. But they don't have their own metabolism. So it just depends on how you want to think of it. I think of them as if ChatGPT is alive or sentient, and that's what viruses are. They're somewhere pretty close. They've got some sort of genetic program going on, and they make a lot of sense to talk about like they're alive, but at the same time without their hosts, they're not fully. They need that help.
I think they're alive.
And I think that's reasonable. Yeah.
Oh, can I ask, I'm very curious about the freezer virus that you mentioned.
Oh, yeah.
Tell the story of the freezer virus.
Oh dear, it's been so many years ago. I think it was an equine, some sort of horse that got infected. It was a lab that had it in a freezer, and they decided to send it to CDC. And the lady who was working on the assembly had to go through this rigorous process where she did basically what my program does, where she would try to blast it, pick a reference, she would assemble it, it wasn't quite right. She would take that, she tried to assemble it again, and eventually she had something out. Then we worked together on this program, and I took the data and I got the same consensus, and I was like, oh, there you go. I saved you a bunch of work if you had that earlier, but yeah, those things do happen. We get samples for different reasons. Most of the samples we get, I would say, are so-called surveillance samples, but there are other kinds of samples we get to. Yeah.
You said there's a lot of lag time between when you decide on a vaccine and when you actually administer the vaccine.
When it is, yeah.
Is there a way to assess if you think that vaccine is going to be less effective? Is there a way to tweak it to get a more effective version? And is there a way to also measure the effectiveness of the vaccine?
So the last question is yes. Routinely, people do so-called vaccine effectiveness studies. So a lot of our EPIs are involved in this activity. Look at Carrie Reed from CDC, you'll find some of her work there. I know she's worked on that. But there's routine evaluation of vaccine effectiveness. Sometimes vaccines are figured out to be ineffective. I remember a few years ago, there was a time where we were not recommending the so-called live attenuated for a time, maybe for children or something, because it was ineffective with the current circulating strains. And later on, we said it was fine again. So sometimes you can come out and say that of the viruses that are available or vaccines that are available, you should maybe pick this one and not that one for data-driven reasons. In terms of actually trying to deal with the problem of the, the time lag, I think people are trying to always make those processes faster, trying to do different kinds of prediction. There's a lot of science involved, but there's probably some art too. I personally only have worked on data that goes into what's called our package. So we have a whole big package from our collaborating center, which is CDC. So I've worked on that data, but I've not actually gone and done the so-called workshop for the so-called vaccine consultation meeting is what it's called. I've not gone myself, but yeah, they're always interested in ways to try and shorten that lag or get a better prediction of what's going to happen in the future. Hopefully that answers your question.
Got a question on Zoom.
Okay, please go ahead. Please, can someone ask the question on their behalf? Garrett?
Just go ahead. You can be.
Is it a chat question or a?
No, it's a, and Abby's got a raised hand.
Oh, sorry, I had to turn my volume up. I could barely hear.
No audio.
Can you hear me?
You don't give her audio.
Can you hear me now?
Yep.
Hearing
Go
that.
ahead.
Okay, great. Thanks for the talk. You had briefly mentioned storing the analytical data, but my question is related to the metadata, especially with something like coronavirus. I'm wondering how rapidly you guys are able to implement infrastructure and what sort of resources you have for resources in terms of both humans and digital resources for both capturing, sharing, and ultimately using the metadata internally.
So I'm not sure what kind of metadata, like collection metadata, laboratory information system metadata.
Yeah, both. So information about any sort of field work or the laboratory work that was done.
Yeah. So unfortunately, the phrase is building a plane while you fly it. This is what they like to use. And so on the COVID side, they were kind of fortunate in the fact that Flu had already done some of this work, but the work and the data streams were different. So we were able to apply infrastructure that Flu had developed and programs that Flu had developed to COVID and collaborated to do that. And so it evolved over the course of the pandemic to now it's sort of old hat, but in the beginning, there was a wild scramble to try and transfer things down. Should it have been that way? I don't know. I work in Flu and we were ready to go. We're always trying to be ready to go. But maybe the idea of a coronavirus pandemic of that time is not something that had been countenanced. So I'm not going to lay blame. I'm just going to say that descriptively, a lot of work went into adapting existing infrastructure. And then those data streams with the metadata that you're interested in, those can exist in different kinds of systems. So getting the right data flow to then integrate that data, it's kind of it's not scientific work really. It's data engineering, but it does take a lot of finesse and a lot of preparation, a lot of thought to try and get it to run smoothly and at scale, because the data just keeps coming, and if you're not ready for it, your system will be able to handle it. So I don't know if that answers your question, but feel free to clarify.
Yeah, thanks.
Yes.
My question is more like career development.
Sure.
So what's the difference of the role as bioinformatician or epidemiology in CDC? Does it take different job station?
Yeah, so for better or worse, historically speaking, CDC has been an epidemiologist first institution. Lab, I would say, has been then the second kind of tier. It's been very important. And informatics, for better or worse, has been sort of, the follower. Because of the pandemic in particular, there's been a surge in all kinds of hiring actions related to data scientists, data analysis, more informaticians. We've always had informaticians that are needed, but I think the awareness has increased how critical that work is, even though, because historically speaking, when you think about CDC's roots with, being in the Southeast, was it malaria? If I'm trying to remember, no, was it malaria? Some sort of vector-borne. Maybe it was. I don't remember. Anyway, but things have gone from very simple routes to very complex and comprehensive public health mission. So that's going to involve data. That's going to involve computer science. That's going to involve statistics. That's going to and biostats. That's going to involve algorithms and traditional bioinformatics, as well as more software engineering types of approaches and data engineering. So that there's a whole broad spectrum of things that you can do with that skill set. I would say that CDC is more mission oriented than, say, probably working in academia, although I've only been an intern and had grad school experience. But your work is definitely shaped by what information products the agency needs to produce. So you contribute to that.
I'll follow the question. So what do you think, like, as a bioinformatician, what kind of quality is lacking compared to the epidemiologist?
So let me try to understand the question. What kind of qualities are lacking from an epidemiologist versus?
I'm a fourth year student as a mathematician. So if I want to make a shift to be an epidemiologist in the future, kind of qualities.
They're just different disciplines, different focus areas. They're both important. And some of them are, more focusing on the collection of epidemiological data. Some of them are more focused on the forecasting and prediction, statistical modeling sides of it. So there's a lot of different things you can do in epidemiology. There's also, I know, groups here that probably study so-called genomic epidemiology and things like that. Molecular epidemiology is probably what you call it. So on the bioinformatics side, that's kind of why sequence surveillance is so interesting to us, is because it combines the traditional bioinformatics work plus some data engineering plus some other pieces with some of the public health epidemiology side. But instead of focusing on traditional case reporting, you're focusing on sequence evolution and the expansion of clades, the expansion of drug-resistant mutations or not, trying to compare genotype with phenotype. So there's a lot of more molecular epidemiology tasks you could do on the informatics side, although you'd probably be working with other people who did more pure epi work. Yeah.
So related question and one more question. So in your experience, is there a blend or is there silos for molecular epi? Like, are epis have you seen epis that do molecular epi, or is it like you have to work together and you guys do that? The genomic side, I mean.
So they do have some crosstalk, but as long as I've been at CDC, there's been this lab epi initiative trying to bring us together. So informaticians typically will work with, if you're an informatician working with the lab, you're a bioinformatician, basically. If you're an informatician working with epis, you're basically a data scientist or a data engineer. Like, that's kind of, if you're going to support, the lab people or the traditional epi people, that's kind of the role that you'll find yourself in. But I've collaborated with them the serology work, for example, that I showed, where we're trying to look at representative strains to see if they're at least as good as the vaccine that I mentioned. That was work that was done by the Epi branch that then we productionalized within informatics, and informatics still does it because we have enough statistical background to do it. But then we could go a step farther and we can apply all the clade annotations to all the sequences, and we can integrate all this extra sequence metadata that we have that the EPIs didn't have when they were running it on their own. So it's just sort of like finding opportunities for those collaborations. But you know, typically the honest question is they've been more separate, but there is crosstalk, and we're trying to bring them together as much as we can.
Okay, and then the other question is, I was wondering how you decided to go from ORISE to FTE versus EIS. Like, have you had experience with EIS officers? Do you know if it was like super necessary?
I mean,
Can we get an explanation of those acronyms?
so yeah, so go ahead.
I was confused,
Oh.
but it's epidemic intelligence.
Yeah. So to me, and I've had limited exposure to them, they're doing a lot more pure epi, whereas I'm coming from, you know, computer science first and math, and then biomedical science, and then mathematics. So I'm coming from a different worldview probably.
Okay.
And so I'm trying to bring, so my particular background from my personal experience, like I'm trying to find math and computer science that I can bring and leverage in the biological domain of viruses for the public health mission that I have. And so, because that's where I lean. I kind of lean towards that computational side.
Yeah.
But there are other people that lean more towards the biology or the statistics. And so if you lean more towards traditional statistics, you might consider epi. Because there's a lot of stats in epi. But there's a whole lot of other aspects to it too. I'm not qualified to talk about
Okay.
EIS per se.
Yeah, I just don't have a good picture of how melding happens. Because from what you're saying, it sounds like it's more collaboration than individuals having like being more multidisciplinary in themselves.
I mean, bioinformatics is multidisciplinary, but
Yeah.
but in terms of doing that, we had an epi in our group, but she didn't stay, who switched to bioinformatics. And she left for personal reasons, but not related to our work. You know, from my experience observing her, she enjoyed the informatics, but I guess I don't know anyone who's, I don't know if I have an anecdote of somebody who went from informatics to epi.
Yeah.
I know the reverse. I've seen the reverse. But if you were to work with epis, you would end up probably starting out doing data engineering, data science, I would imagine. If that's, because you could use your same skill sets in programming and hopefully statistics to leverage yourself that way. Data visualization.
Yeah.
All those things are important. And the epis are not necessarily, they're not usually very computational.
That's kind of what I was wondering.
They're using SAS, R maybe, and you know, they know what they know, they're experts, they're to be respected, but that's why there are other positions that can augment their function. And they get informaticians too. They will hire informaticians, but I'm saying the role that they play is more of a data science, data engineering.
Yeah.
Yeah.
So her hired as a bioinformatician into an epidemiologist group at CDC, like would they extensively train you to kind of start using like the same kinds of analyses they use, or would they bring your own individual expertise into their group?
I can't speak for everyone at CDC. But my own personal experience watching others is that there's usually some sort of cross-pollination where there's particular project or report that they need to do. So I can think of some data analysts that I know, data scientists or data analysts that I collaborate with that are very good, that work with the EPIs. And so they're probably bringing their Python programming and their data visualization background, and they're helping the EPIs solve their problems. So it's more like they're bringing their skills to their problems. So the EPIs are providing the data domain, whereas the informaticians are providing the missing skill sets for that data domain. I don't know if that answers your question. That's how I imagine it. Again, like your mileage may vary on depending on who you are with. Could be different in different groups. Small anecdote. Any other questions? Online?
Thank you, Sam.
All right, well, thanks very much. Those who are sticking around for lunch, stick around. If not, you can. See you later.
Cindy, we have to follow that.
Everything has to be exact.
So you just.